Hi everyone, welcome back. In this video, we're going to keep working on our BioSeq class. In our last video, we have created a new BioSeq class and started transferring all of the DNA Toolkit functionality into it. So here it is. As you remember, we have created this initialization function that is part of every class, and it just does the validation check for us. It makes sure it's all uppercase, creates a label and sequence type, and validates itself to make sure that we are working with a valid string. We have transferred our first two functions, the validate function and random sequence generation function. We have also created these new two functions, bioseq type and sequence information. These two just return the information about the sequence that we're working with. We have new file structure here, bioseq.py, biostructs.py, and main.py still is our test file where we test our code. So let's run it again to just refresh on where we left off in our last video. So if we're going to run this, we can see that we are shown this information about the sequence. So we create this instance of our bioseq class here, and we generate 40 character long sequence, which is DNA, and we're just printing out this information right here. So let's do the same thing as in our last video. We're going to look at our old code here, side by side, and we're going to be transferring old code into our new bioseq class here. Let's get rid of all these windows here too. So we have transferred validate sequence function. We can actually delete this here so it's not there anymore. So the next function we're going to transfer will be nucleotide frequency function. As you remember as well, we're not going to be just transferring functions, we're going to try optimizing them. And luckily enough, we already did most of that job in our last videos. Here we had this more Pythonic approach, which is commented out. So let's cut this function out and paste it into our class. Of course, we need to nicely tab it like this. And get rid of all of this code here. We're going to use this counter method. So we need to do, of course, a couple of changes here. As we remember, we're not passing anything to our functions anymore. We are working in a class. So class will perform functions on itself. Can you count nucleotides in yourself? So instead of a sequence, we're telling it count nucleotide in yourself. And then as well here, we need to add self. And it's saying that counter is not found, counter method, because we did not include the collections data counter from collections import counter method. Very useful method. Now we see there's no errors here anymore, and we're going to keep our doc strings as well. So right away we're going to go back to our main.py file and test our new function. Not new function, but newly transferred function. So nucleotide frequency, it's there. We're going to save, and as you can see, we're not really passing anything to these functions at this stage. Let's run it. And we can see that it returns our dictionary with the count. Great. Next function is transcription. We're going to cut this from here too. We're going to paste it into our bioseq class. Here we are doing the same changes because we're working on our cells. We're going to say self, and we are going to say here self2 because we need to return a replacement of T and U on itself. So one thing to note here, we are not modifying our original DNA sequence in here. So when we create our class up here, we are passing it a sequence and we store it in self.seq. So we pass some DNA string. As we can see here, for example, this is a DNA string we passed to it. It's randomly generated, of course. And here we're just saying, can you return a copy of that string, but replace all T's with U's? This will not, again, modify our original sequence. Our functions are going to return a copy of whatever we need to do with this sequence. In this case, we're doing transcription. So in this case, we're telling it to return a transcripted copy of our string, not transcripted string, a copy. Okay, let's save this and test it right away in our main.py. Let's copy this output. And let's see. Okay, it finds transcription. That's a good sign. We're going to save it and run it. And here's our RNA. Just to prove the last point, let's try printing our sequence again, just to make sure that when we ran the transcription function, it did not modify our original sequence, because that would be really bad. So we create a sequence, load it in, it's a proper genome, and then we do some functions on top of that, and it modifies our genome. So it's going to kind of mess up the whole workflow for us. We need to make sure that this is not modified. If we want to modify this somehow, we should create a new instance and do modifications on this original string we loaded and save it into a new variable, okay? So let's do a quick thing. Print test seek. Okay, let's see if it changed. So, okay, yeah, we can see it did not really change. That's the original string. We can select it and it will match the similar string. So it matched those. So again, we can see that the original string was not modified. That's great. Let's go to our next function, reverse complement here. And reverse complement 2 has a Pythonic approach. We're using a string method make trans here. So let's cut this function again. And let's paste it into our Bioseek class. For some odd reason, it doesn't like to tab it correctly when we paste. So we're going to do that here. And we're going to modify it as well. We're going to pass a self because it's reverse complement on itself. And we're going to get rid of this list comprehension. We're going to uncomment this. Some cleaning and some other modifications. We're going to insert self here because we are doing the translate on itself. We're not going to use self here on the mapping because this mapping is only used in this function. It's local to reverse complement function, so we don't really need to specify self. So we have discussed this functionality in our previous videos. So string method make trans just creates a translation from one sequence to another sequence. And in this case, we are returning again a copy of our string and we are applying a mapping to it and we're reversing it right here. So it is a copy, not the original string. Let's save this and go back to our main.py and run our test. Okay. Reverse complement. Here's our reverse complement. So far so good. Next one is this small function GC content calculation. Cut it out and let's paste it in here again. Couple of modifications as we know by now. We need to GC content calculation on itself. And here as well, we need to run self seek count. And one more modification here because we're counting the length on itself. That was a quick modification. And now we're going to do a quick test as well. GC content. 50% of the string is the GC nucleotides here. Okay. So the next function is a bit more tricky. We're going to have to modify some of that. We're going to cut it and we're going to paste it in here. Of course, we need to tab it. Let's add self to seek here as well. Okay. And this part here as well. And we can see one problem right away here. So GC content is here now, but it's even if we're going to do self, okay, self, it's still going to complain that it does not accept any parameters. It only works on itself. So one thing we could do here, let me make this a bit bigger here. One thing we could do is reuse this functionality. So if we're going to copy round and paste it in here, like that. So it's self-sustained function. But we need to change this self sequence to this subseq because we're not working with the sequence anymore. We are creating a slice of our sequence and we are working on a subsequence here. So if we're going to do this, in this case, we're not going to need self. And in here, we're going to replace the whole thing. In this case, we're not going to use self because again, subseq is local to this GC content function. This variable here is not used anywhere else. It's being created here. The slice of our sequence is being put into it. Some operations being applied to it, and then it's being discarded. So let's try running our code. Okay, let's test it. So we have GC content with subseq, and we just gonna run that and use a default value. Our sequence is of a length 40, and that's split in two. And the first 20 characters, we have 55% of GC, and the second part here, another 20 is 35%. Okay, so this seems to work so far. Now we're gonna go for our next function, translate sequence. Gonna copy that too, paste it in here. Okay, so typical modifications, we're working on ourselves. We're going to keep this init position. Of course, we need to modify this here as well. Self.seek. Okay, now we need to use our DNA codons. So we're going to go back to our old structures file. Let's move it to our old code side here. And we're going to use our DNA codons. I'm going to copy it and paste it into our new structures. So now we have to include that too. And instead of including everything, we should follow something called PEP8. It's a Python standard of writing code. I will include a link if you want to have a read about that. But we're going to include DNA codons, and we're going to use DNA nucleotides too. We have to specify what we're going to use from that file. So now we're going to come back here, and we forgot to modify this part here. Our editor helps us in this case too. So I don't see any more errors or complaints. Let's go back to our main function and try running this. Copy this output. And we're going to run translate sequence. We're not going to pass initial position. Let's do this. And of course we get our amino acid sequence right here. That's great. Our next function is Codon Usage. I'm going to cut it, paste it into our class again. Okay. Now we need to modify it to use self part here instead of a sequence. And by using a code editor, we're making our lives so much easier because it's actually highlighting the parts we need to modify. We need to modify our sequence, of course. Okay, it doesn't seem like we need to modify anything else here. Let's test it right now. Codon usage, and we need to specify a codon we want to see used in this function. Let's try running a couple of times. Okay, we're lucky from our second try. Here it is. And here it is here. So this function works too. Our next function is generating reading frames. Now here it is in our class. Let's do the tabbing first. And we're going to see we need to modify this function quite a lot. So we're going to do the regular self stuff here. Okay. Because translate sequence is not a function anymore, it is a method. We're not passing anything to it. We're just saying self translate sequence, and we are passing 0 to start from 0. Okay. So we need to add this to our two functions here now. Let's delete this and paste it in here. And of course, we're going to set the offset 0, 1, and 2. Okay. Now we see another problem. Translate sequence doesn't really accept anything but initial position. But in the original code, we said translate sequence, we were giving it a sequence and a position. So this is not going to work. In this case, we need to create a temporary sequence, a copy of our sequence, and run the same functions on it. So here we should create a temporary sequence, tmp-seq, temporary sequence, and we're going to initialize our class here as well. But instead of initializing it with a sequence, original sequence, we're going to initialize it with a reverse complement sequence. So we're going to say self dot reverse complement. So this is going to take this original sequence that we have here, reverse complement it, and as we remember, this will return a copy. Okay, so it returns a copy of our original string, but reverse complement of that string. We also should pass the type. This will be needed when we're going to add RNA support. We need to make sure that when it creates a copy, it's a DNA or RNA. Okay, so let's modify this part here too. We're not going to use this whole code here because we're working on our temporary string, tmp-seq dot translate seq. Okay, and we're going to do 1 and 2 here. We are not using self here, again, because this is local to this function. And a good idea is actually to clean after yourself. So we create the sequence right here. We apply some operations onto that sequence. And by this line here, which is 93, we have our list populated with needed reading frames. We don't need the sequence anymore. So we can help a garbage collector to pick this up and free some memory for us. tmp-seq. We're going to say delete temporary sequence and just return frames. Okay, so we don't see any complaints anymore from our code editor. So let's try running this function. Let's copy this. generate reading frames for us. Let's try running this now. Okay, we can see that this works. What we can do instead, we can do a loop because it's a list of lists, right? So for rf in test DNA gen reading frames, please print rf for us. Okay, so that's a nicer format. So here are our reading frames. Okay, let's go back to bioseq. Our next function is protein from reading frames, where we scan our reading frame and try to extract a protein from. Let's copy this here too. So now the only modification for this function is we're going to add self to make sure it's part of a class. Okay. So the only modification we did in this function is a self because it's part of a class again. And this function is not really used by us as a user. It's used by the next function. We're going to transfer this, all proteins from all reading frames. So that's the only modification we did. We can still try testing it. We can generate a reading frame like that, for example, copy it, and go back to our main.py file where we test things. And let's print test DNA.proteins from reading frames. Let's paste our list into it. We're going to create this list. And here it is. Let's make sure it has a start codon and a stop codon. Like that. And let's run it. Yes, we can see that our function works and it generated some sequence right here. So the last function we're going to transfer is our all proteins from reading frames. So we can see there's no functionality left. Cut this, go back to our PySequence class and paste it in here. Of course, we need to tab this. So now we have this pasted in. Let's see what modifications we need to do. And there are quite a few. So the first, of course, is self, because we're not working on a sequence anymore. We're working on ourselves. The first problem we see right away is generate reading frames is now part of this class right here, and it works on itself. It generates reading frames for itself only. It doesn't accept every parameters, but here we're saying, can you generate reading frames for this sequence? I suggest we take the same approach we took here. So here we're going to create a TMP sequence, a temporary instance of our class again, bioseq. We're going to instantiate it with the slice we have here. So of course, self. We are giving it a slice of our original string, and we're going to specify a seq type as well. Again, this is useful when we're going to be working with RNA as well. So we're going to add RNA support, and this is going to take care of differentiating between DNA and RNA strings. And on the line 1 to 2, we need to modify this to do this. TMP, generate reading frames for us. And that's it. We're not passing anything to it, right? Because it's generating reading frames for itself. And here we need to do the same thing. So we're going to do self.generate reading frames and get rid of this. Just a couple more here. We need to do self here because we are generating proteins from reading frames on our cells, and we are giving it a reading frame. So we can see that proteins from reading frames accepts an amino acid sequence, and that's what we are providing. So it doesn't seem like we need to do anything else here. Let's try testing our code. We're going to add this output here. Test DNA. All proteins from all reading frames. Let's get rid of this out because it's confusing. Okay, so let's run this function right now. And that's where, remember, the rules are we have to come across a start M codon and a stop codon to generate a protein sequence. So let's try running this a couple of more times. Maybe we can come across some random sequence. Okay, here it is. So MRAI, and there is a stop codon. Okay, so we have successfully transferred our functions into class methods here. Of course, you can go ahead and expand this class as you wish, just to fit your needs if you're going to reuse this class anywhere else. We can go back to our file structure right now, and we can get rid of DNA Toolkit. We don't need this anymore. We have a bioseq class. And we can actually get rid of the structures.py file too, okay? For now, we have just these two files, bioseq and biostructs, okay? We are going to be expanding this a little bit more when we're going to add RNA support. And our class is going to change a little bit. We are going to add a basic logic to check if it's an RNA or a DNA string we're working with. So some of these functions, for example, transcription, we're not going to apply transcription to an RNA string. So we will need to add a logic saying it's not a valid DNA string. So I cannot do transcription on here. When we're going to be doing translate here as well, we need to make sure that we're using DNA or RNA codon tables too. But we're going to take a look at that in our last video, which is our next video. Okay, so this is it for this video. Let's just try running it again to see all of the functionality working, and it's all wrapped into a nice class. Now you can include this file into any of your projects, extend this class as you wish, and reuse it anywhere you want. So the basic idea of the series of these videos was to show you how you can take very simple biological concepts and transfer them into the code. In some cases, some biology students are asked to write some code and use something like BioPython, for example. So they go in and they look at some variables, how functions work, some loops in Python, and then they include BioPython and start working with it. Of course, you can include any module and start working with it, look at the documentation, just take a look at what parameters function accepts, what it returns, and work with that. You don't really need a deep knowledge of Python. But there are cases where you are giving some unformatted, dirty data, and then you need to parse it, you need to clean it. Maybe you need to write some custom code to process that data or do some other functionality that your module, BioPython, does not include. Then you will definitely need a medium to advanced kind of Python or any other programming language knowledge. So that's what we did here. We built a small class that can be reused. We tested it from scratch. That should give you an idea how you can build your own tools on top of existing tools. So where do we go from here? You can take this class and reuse it in your own project, as I mentioned. We are going to be working on some genome analysis class as well, where we're going to be reusing Bioseq heavily. We're going to be modifying it as well. We're going to be adding some extra functionality as we need it. I'm also working on a GUI, which is graphical user interface, on top of this Bioseq class. So you don't have to run Python scripts. You can open just a nice app that is cross-platform, runs on Linux, Windows, and Mac. It's using this cute library that is cross-platform. So this just to show what you can actually do if you are interested in developing software for bioinformatics. So here's a very basic example of the application you can do. It's still an empty application for now, but it's a UI user interface built on top of our Bioseq class. So what we can do here, we can try adding a sequence, we can try loading it, and it will say exactly the same thing as we've seen in our test output. We can of course try loading nothing, it will do some checks, no background, no sequence provided. We can always do this and try importing RNA. It's going to say an RNA, a GCT, which is not RNA. It should say that incorrect RNA sequence provided. We're going to be adding these checks in our next video as well. You of course can load from a file. Let's just load from a file. You don't see that window, but I'm going to load, for example, Corona. Okay, so here it just loads the sequence. Okay, we can just load it in. And it complains that it's not an RNA, of course, because it is a DNA. Let's try loading it again. And here it is. Okay. Label has been created automatically because this supports a fast file format reading. You can add all of these interesting features to your set of tools. And this Bioseq class that we have created and completed is not just a small piece of Python code. You can be reusing this in your project. You can build a UI on top of that. You can do a web version of that as well. But I'm going to talk about this in my upcoming videos. And one last thing I want to bring up today is Rosalind challenges. As one of our community members correctly brought up, we should not be sharing solutions to Rosalind challenges. That's part of the rules of Rosalind. So from now on, we're not going to be explicitly looking at how to solve a certain problem, but you might be seeing this symbol you see on the screen right now from time to time when we work on our stuff. That would mean you could use this code that we are currently working at this part of the video to solve some Rosalind challenge. So it will be up to you to see what can be solved with this particular piece of code and how to modify it to actually pass the Rosalind check. If you still can't solve some particular Rosalind problem, you can always join our chat and we can discuss how we can solve a particular problem, hopefully not just sharing the solution, as Rosalind, again, prohibits that. They are asking for users not to share solutions, because this is the whole point of Rosalind, right? People join, people try solving some problems, and that's how they learn. Okay, so this is it for today. Thanks a lot for watching and listening, as always. And I hope now you can see how you can use very basic Python functionality to build something like a Bioseek class for yourself. Extend it, reuse it in projects, build some graphical user interface on top of that, build some web interfaces on top of your code, and basically reuse it in your own projects and understand what is happening underneath the hood, for example, if you use BioPython. In the last video, titled DNA Toolkit, we're going to complete our work on that DNA Toolkit class by adding RNA support. We're going to add read and write files functions, and we're going to add our FASTA file reading function as well. So our DNA sequence class will be complete. We can be loading any text into it, we're going to be loading FASTA files into it, and it will automatically pick up and understand what kind of file it is. We're even going to add an URL support, so you can give it a URL that points to some text file with some data, and that can be read and understood by our Bioseek class. So it's going to be like very diverse class, and it will report all the errors if it can't load some particular sequence instead of just crashing. But this is going to be our last video, number 9. So until next time, Rebel Coder signing out.